So what I had really sort of envisioned for this was going beyond some of the efforts that are being made with these other breakouts, where ontologies, sort of assuming ontologies have been defined, and that we have some common framework to talk about cell types and so forth, to think about this other challenge, which I think is actually quite formidable, of trying to bridge data types and scales, where, for example, we are going from the perhaps subcellular, certainly molecular, up to cells and up and onward out to tissues, using the plethora of tools that are coming of age and have already been deployed for some of these atlases, much of which we talked about yesterday. So these are new spatially resolved transcriptomic, proteomic, and genomic approaches. Of course, the dissociative single cell methods and our classical genomic approaches, and how do we start to integrate both temporally and sort of in tissue context. And I think a lot of this comes back to the tissue context. But I'd love to hear, now that we have a sort of a smaller group here, smallish, actually quite large group I see, for those that are really keen to see this go forward. Certainly Ian, I know you've been commenting on the thread, and I think others as well, but to hear what else you would like to see covered. Welcome, Peter.
I mean, Christine, I'm interested when you mention those tools as to exactly what kind of tools you're talking about. I mean, I think there's a connection in this with some of the discussions some of us were having yesterday about provenance. I don't love that word. It's a bit of an abstract word, and connecting it to biology can be tricky, but I think simply, I think it's the glue that grows across the levels that you're talking about. If we needed to explore that idea, perhaps we could at some point, but I think it's more interesting to go from what you're talking about in terms of tools. And I think you're probably talking about tools that tell us something quite directly about biology and that are going to be analytical in some sense, and are going to give us biological insights. I think all the provenance stuff is plumbing that stuff needs to run on. But I think it's important to focus on those tools. So if you could say a bit more about that, that would be helpful.
Sure, yeah. So I think there's a variety of techniques that one could envision, and it's really not mutually exclusive to one or the other, because we're talking about these very diverse data types. I mean, I think on the one instance, we're trying, at least how I view some of this problem is, how do we start to assimilate this information that's coming from disparate cell types? So maybe multi-agent-based models where we're actually simulating in 3D the tissue architecture, cell types being agents in this model. And we've now encoded that information about cell types, for example, from some of these high resolution technologies that have been built. So that's sort of on a high cell type tissue architecture scale. Of course, I think a lot of the measurements that are requisite for really building certainly realistic models are just coming of age. But the nice thing about these kinds of approaches is they can also encode dynamics, such as lineage information and the like, which I think is an important facet, certainly the temporal component of many of the atlas efforts that we're engaging in the tumor space, which is either we want to understand something about that cell's past proliferative history, or indeed we might actually have some temporal resolution. So that's one flavor of models. There's also, I think we don't want to get too into technical components of modeling, because right now it's much more about what are the sort of types that are, types of approaches that are relevant here. But, we also have this fundamental challenge of just integrating heterogeneous molecular measurements. And by that, I'm really focused on the molecular scale and saying, okay, we have perhaps single cell RNA-seq, single cell attack-seq, maybe bulk attack-seq, as well as some kind of spatial proteomic measurement. What are the types of models that allow us to think about putting together these data types and jointly learning on them, as opposed to taking an output of one And I think, there's some powerful approaches in the machine learning space that allow us to think about this. I mean, multiple kernel learning is one, but there are others. And that's a different class of integration that doesn't necessarily link back to the cellular yet, right?
Christina, I was wondering, sorry, this is Peter Sorger. I'm
Yes.
just interrupting you, excuse me. Picking up on Ian's point, but also I think what you're saying, it seems to me there are some fairly basic issues here that I wonder where they're getting resolved. And you even find them in, simple cases of data integration in cell lines, right? Which is, just to give you an example, we have an activity, NF-kappa beta, that, drives gene expression that we can score and model, but it's made up of some combination of two proteins which have at least 6 different genes, right? And I think, it's a sort of prosaic formulation compared to what you said, but it is true that, you see the upregulation of the subunits, but the integration of those into a functional definition of active NF-kappa B is missing in most cases, because even those mappings are largely missing. So I always find that these discussions tend to get quite sophisticated, but even some of these elementary mappings between protein and gene space and function space. And then of course you get into a cell case, you've got ERK and it's 2 genes and they can be in the cytoplasm and the nucleus. And often those relationships are undefined. And I've just wondered if you or anyone else has started to do that even for subsets of genes.
We've done it for subsets of genes on us, and I agree this is a really hard problem. We've done it for some of the chromatin regulatory genes with particular, biochemical questions in mind. I think that this needs to be done much more broadly, as you say, and I'm actually really curious and would like to hear about some of the approaches that you've used from the sort of system pharmacology perspective, where maybe the measurements are different. And I think it's, again, you're touching on a really important point, which is the integration of in vitro measurements, cell line assays, maybe trying to map that back onto what we're seeing over very coarse time series data, potentially in tissues. And so, how do we bridge these temporal components as well, where we can make detailed studies in the context of perturbations?
I don't think there's any simple answer. One thing I'll say and then open it to everyone else is that in the context of machine reading of text, we're trying to figure out what exactly, P53 or something actually, well, that's an easy one, of course, but, P65, what is it? We did build a tool we call FAMPLEX, which is families and complexes, that tries to define individual HGNC terms in sort of functional world. And rather than go into that, I think what we observed is there's a really interesting conceptual aspect of this that tends to get missed in the technology, if that makes sense. Ontologies and things, people their eyes go to the sky and they get raised. But there's a very interesting question of, which genes does it need to be present in order to get a functional molecular complex, that I think is a lot more interesting and is really the foundational piece of any kind of ontology of this type. But when we tend to discuss it, we tend to get into the weeds of discussing ontologies. And so it's almost as though that field is undefined. The whole aspect of the molecular basis of cross-platform integration is only poorly developed.
I may say something about this representation in ontologies. I work in ontology development, and there is this, there's been...
My apologies for saying something.
there's been an effort with the protein ontology to try, and I've been involved in the specific representation of a pathway, toll-like receptor pathway, trying to represent all the events, starting from the ligand binding to the receptor and all the conformational, all the change, all the assembly of different units of different protein or complex in order to represent that. So the protein ontology is trying to do that, and in fact, we have defined a protein complexes, which each protein complex has the complexes and has how the complex form. And I think now they are trying to link to each subunit and even to the complex, the functional activity. So when, and this process, the work that we have done as a prototype with toll-like receptor was really going from the surface of the cell inside the nucleus to the activator factors, and in order to represent everything. So I think it's from the ontology point of view, something is trying to be done. It's not easy because obviously each of the functions change with the context. So it's not, and then sometimes even depends on the quantity that obviously the ontology never represent. So that could be another issue, but I think you're right. That is something that needs to be focused on. And I would just speak from the ontology side. That is something that is being started.
That's great. No, I think that's a really important point. I guess I want to make sure that was captured by our scribes and then ask everybody, we have a group here, we're supposed to be taking a break. I don't want to hinder your coffee break. So I guess maybe we should take a brief pause and come back. I also don't want to lose people along the way who disappear with their coffees, if that's tempting. So, you know, I would just say maybe we should pause for a few minutes and do that if people need to, or I'm happy to keep talking. I don't mind. I've got mine here.
It's a pity because it took us so long to get here that.
Yes, well, I'm fine to keep going.
I think it would be good idea to keep up some momentum. I probably over caffeinated earlier anyway, so.
Sounds good. If people are happy to keep going and others need to step away, that's fine. I think this is a really good dialogue, and I'd like to hear from as many as want to speak up on this, on the challenges, because I think they're actually quite diverse. And I will say, we struggled a bit in framing this because actually I believe that there's so many challenges on the integration level that it was hard to even get to integrating across atlases when we're still wrestling, quite frankly, with how to integrate within one atlas. And so that was a sort of a talking point for how we were trying to tease apart these different themes. But it sounds like we're hearing about ontologies and having common standards for that. And I know, you know, to some extent, this also touches on what I believe Joe was trying to tackle in terms of some of these operational definitions that we have around what are the types, what are the cell types, what are the intracellular relationships that we want to be capturing here. I think a key point that I see when we think about this in the context of integration is context matters a lot. And that's, I think, came across in what you were just describing, Anna Maria, and Peter as well, right? I mean, context is everything for thinking about these molecular machineries. And one of the, at least, challenges that I see is trying to port information that comes from a very specific system over to interpreting that function perhaps somewhere else. And I'd be curious if, yeah.
So, Christina, can I ask a question? I mean, and it follows on from the ontology point, which is, besides ontologies, I mean, I'll start here, which besides ontologies, there's something which complements them very well, particularly with this question of context that you just mentioned, and that's data models. I mean, models is one of the names that you had in the description of this session, but models here can be an overloaded term, as they say, because in parallel to this, there's a whole session, and I looked at, is that the session I should be in, about data models? But in fact, I think what they're talking about is the mathematical models, of biology, which are great, which we work on with the data. I mean, so how are you thinking of models in the context of this discussion, and which kind of models are you talking about? Perhaps both, I suspect.
Perhaps both. Yeah, I mean, I think we actually started out, quite frankly, with a lot of these concepts and then said, well, you know, there's sort of, yeah, I mean, different layers to these models. So I think in order to move and to really have a proper integration of the, you know, certainly across these data types, we actually need to have good models for some of these lower resolution problems. And by that, I mean models that capture gene regulatory networks, right? And cell-to-cell signaling. And that's a different
model of a gene regulatory network is a different thing than a data model for provenance.
Correct.
That would enable this. And I think it's important that there's definitely an interconnection between them, but there's a different skill set necessary to do those and a different tool set. And they probably need, in the separation of concerns way of thinking, need to be thought about as separate concerns.
I think it's the linkage between the two where the excitement area is, right? We're starting
It
to
is.
get, and actually, I think this, to me, is one of the why I'm here. I'm more on one end than the other. I'm more on the sort of agent-based models, where I worry about parameter space and creating models that take the life of the universe to actually properly parameterize. But I am very interested in the data, and I do actually I'm involved in the data collection, but I haven't really come up with a really good way of linking all the human cell atlas data that we're generating in the group to actually the mechanistic modeling other side are all physicists and mathematicians. And for me, that's, to really get the power of the data, exploit it properly mechanistically, we are going to have to link the two. And that's kind of why I sort of decided to jump into this group.
Yeah, I mean, how I think about that is that the biology drives the model. Now, there's a problem when you talk about that with a data engineer. There can be a problem. I think
speaking of
as biologists, we need to take some ownership of these models and to understand how they work, because I think, for me as a biologist by background, I learned how powerful they were in order for me to do things biologically. I didn't get into it because it was fun from an engineering point of view. I think, as biologists, we need to take some ownership and engagement in that, which means a little bit of understanding the skill set of doing the models.
Sure, and I guess, Ian, if you could expand on the types of models and applications that you've been doing. It seems like we have a fairly diverse representation here, maybe not surprisingly. And even if we were to talk about modeling at large, it's actually hard to get, there's all flavors and varieties for this. And I think some of us are coming maybe from a more mechanistic perspective. It sounds like that's what we were just hearing right now. That's certainly, I guess my work bridges the mechanistic, where we're asking questions about evolutionary processes and trying to build in molecular data to that. And there's certain types of, molecular data that are more easy to integrate. To give you a concrete example there, and maybe we can go around and talk about this, because I think it touches on the question of the types of approaches we need if we're thinking about mapping lineage, right? This is tumors evolved through an evolutionary process, so we can use those point mutations as a readout of what has happened, and we can layer on mathematical models onto that, so like a stochastic birth-death process. And if we want, we can embed that in an agent-based model that then keeps track of the spatial relationships of those units of division, the cell. So the cell is the unit of division, but the molecular data tied to that is a genotype, a mutational state, a copy number state, perhaps even epigenetic, right? And then you can embed this in a cellular automaton model. You can keep track of spatial structure. And we believe that you could start to embed multiple cell types in these models. So not just tumor cells, but the immune, the stroma. And in some sense, this gives us some handle on what that process looks like. We can measure parameters from that, but it has to be coupled with other statistical models. In and of itself, that agent-based model isn't going to allow us to infer these parameters that we like. We have to then couple that with, in this case, a model that is adapted from population genetics, so approximate Bayesian computing. And now we can measure back parameters from patient data from this simulation, this virtual tumor, if you will. But I can say that one of the areas that we've struggled with is how do we bring in all these other molecular measurements that we would like to see linked to this now that we can make them, and how do we bridge the cellular signaling scales that are taking place alongside that. And I don't have a good solution, but I'd be really curious to hear for those that are thinking about models to address biological questions, what those are, and if we think that if there was a common framework, how we might intersect them, because I think that solves a big problem across.
One thing I've seen, Christina, somewhat in picking up on what Mark said and Anna Maria said is, so my lab does, we're a mixture of computation and modeling and biological measurement, mainly mechanistic modeling. And what's sort of interesting is what's well known, and I think Mark alluded to this in mechanistic models, is say of cell signaling systems, is that you don't need very many species, individual proteins, before you have an enormous combinatorial explosion in the number of subcomplexes. You know, famously there are, you know, 10 to the 8th EGFR receptor subcomplexes, even when there are only 10 to the 5 proteins being expressed. And so the issue of an ontology and the issue, that sort of conundrum for biochemistry, they actually intersect with each other because you can't fully enumerate. You can only statistically estimate. And so, you know, ontological concepts tend to be hierarchical and enumerative.
Yep.
And I think we've realized that that's not possible technically. But, you know, underlying that technical inability to model every subcomplex, I think, coming back to your point, is beginning to understand what the abstraction layers are, because, you know, successful large-scale modeling like pharmacokinetic or pharmacodynamic modeling has sort of built in a series of abstractions that allow you, as in a multi-physics problem, to ignore some of the details while you take on the detail at a different scale. And I just wanted to put a question out to the group. You know, what I would say within HTAN, which is the Human Tumor Atlas Network that we're in, the observation I would make is modeling of all types is vastly there's a vast underinvestment. You know, the whole program is really, and I say this as a person who grew up as a biochemist, right, is largely focused on data collection. And even though we have, I can't count how many people, we have 27 people in our room, as a whole, it's a small group of us that continue to get together on this. And so I think one of the reasons why these problems are hard is that they're actually really big problems and they haven't been subdivided sufficiently or with enough people working on them. So if you look at the number of people talking about the things we're talking about today compared to, you know, what the intensity of effort that went into genome assembly or something like that, it's very small. And a lot of these large efforts have had the same thing. You know, famously TCGA took them a long time to get around to some of the data modeling. So you know, I just think that, you know, one of the almost trivial observations is that there's actually a need for these problems are hard partly because they're very big. They haven't been subdivided very effectively. And I would argue that's partly because there's not much investment in them.
Can I just jump in and say something facilitatory here? And so it's 11:15, so this is when we should be coming back from a break. So I think we need to start thinking about moving to the second half of this, the solutions. So maybe we should sort of recap what our challenges are and try and pick some that we should find solutions for.
And another facilitatory thing is, I mean, Toby before he dropped in and dropped out, but one thing he pointed us towards was the Google Doc. I put one, I don't think, I mean, in some of the sessions, I don't know how you want to use it, Christine, but in some of the sessions that I was in yesterday, they were very active and everyone was kind of contributing in them.
So we're
I can paste it into the chat.
Yep, it would be great if you paste it. Thanks. I had it open here, but I wasn't sure if you were just updating anything there, Sean, or not. But anyone's welcome to add to it. And I would hope, actually, I mean, I think this has been a really great discussion so far, just bringing to the forefront some of the issues. And if we could nail down a few of the core concepts where there might, in fact, be, you know, a further collaborative group that talks about some of these issues, I think that's really one of the goals of these breakouts. And I, for one, completely agree with Peter that this is underrepresented. It's actually, I find it hard to find people to talk to about these types of models and questions. And it is really a niche area unto itself for each of these different questions, the different data types, because there's a lot of complexity and it takes having the data in hand first. So I agree we should move towards the questions. But if there is anybody else that's here that wants to share just a little bit of perspective, maybe on the types of models. Mark, we heard a little bit from you, but I'd love to hear more about the questions that you're actually tackling. It might help us narrow this onto what we can do as a collective, or, you know.
But I'm an immunologist. Sorry, go ahead.
No, please go. Okay, sure, go.
I'm an immunologist, and we're really interested in how we model immune responses. I think the biggest challenge, so I'll just give it very brief, is getting biologists to have confidence that the models are useful for them. It's slightly easier when I go into doing stuff with pharmacologists, much more difficult to get immunologists to, they just about believe the bioinformatics, but they don't believe mechanistic models yet. They lack that confidence.
Yep, I agree.
Yeah.
So maybe I'll just throw in my experience. This is Fiona Ginty from GE Research. We've been working mainly on protein multiplexing, but we have a ORA1 on colon cancer at the moment where we're trying to bridge cellular measurements of apoptosis with a differential model that's been developed by our colleagues in the Royal College of Surgeons in Ireland, where they've developed a prediction of apoptosis potential at least before we started working with them at a tissue scale, and now we're bringing it down to cellular scale. And so I think we're not there yet. We're about halfway through the project. We've generated all the cell data for the apoptosis markers. They're feeding that into their model. Controls were essential, of course. We have controls for every one of the proteins at different levels, so the model is correctly calibrated. But yes, there's a lot of learning from both the modeling side as well as the biologist side. I'm the biologist amongst the rest of my team. And we have to have, that sort of validation, constant validation along the way, either independent measures, RNA, tissue level measurements, to ensure that the types of readout we're getting from the model relate back to what we, what's being predicted and what we measured at a single cell level.
Yeah, that's very helpful to hear, and I think actually touches on some of these other issues of going between these heterogeneous data types and trying to ask a specific biological question from the data. Yeah, so this is great. I think we're hearing a lot of the challenges, and they hopefully resonate with many. I mean, one of the questions here is, how do we, how do we define, I mean, some of this comes down to, I think, the questions that we're trying to ask, the very specific biological questions in the framing. And Ian, this is something that you've touched on a few times, is having a biological lens on the question. And I mean, there's a huge swath of things that we could be addressing, and the types of, depending on the biological question, the data types and the type of models that are required are going to differ. Which is why I think we're in this very large space of problems here that span.
Can I say a little bit about it, Christine?
Yeah, please.
So, I mean, you sort of planted a seed about it earlier, and I'm thinking about it a bit. And, a couple of things. One is, if I think about coming out of the biology lab and being told, yeah, Ian, you need to learn data modeling. So I went to data modeling class and I learned, well, what is an entity? And an entity is a thing in a data model. Depending on your religion, data modeling religion, and there are different religions, but let's go with this one for the moment. An entity is defined in that data modeling class as a thing of significance to the business. Now, I was learning this at Oracle, all right? So the business meant business, but our business is science, and our business is biology. And so how I translate that is, an entity that things you want in your data model are the things that are significant to biology. So you would end up with a data model for biology that has things like cell and, well, I can't stop there, right? We know what a rich world we live in. The other thing is that, I mean, and I still use it, which was a warning from the guy who sort of was a guy called Richard Barker, who was the inspiration behind, and he actually worked for Oracle. But his message was around, because data models can get very generic and abstract. And his message about it was, these models are a communication vehicle, all right? And if your model is not communicating to somebody else, and even what I'm saying here sounds very abstract, it's not doing the job that it's supposed to. To try and make that real, to try and make that specific, is if your data model, and I'm talking about the data models that we would get if we go and look at things like the GDC, Genomic Data Commons, and TCGA data, I mean, that's where you would start, but it gets more complex from there. If you can't go to those and understand biology, then it's not going to communicate what needs to be communicated. I mean, the example I've used recently is a pancreatic organoid study that's in the Genomic Data Commons. And I can go and see in that there are about 10 specimens from a given subject. And I can see that 4 of them come from liver. But the GDC doesn't tell me that 2 of those came from the same metastasis and the other 2 came from another metastasis. And that tells me something that I need to know to do something useful from a biological point of view. So what got lost there, I'm going to say, was communication from the person who designed that experiment as to what the data meant. And so the model failed to communicate that. Now, there's only so much time in this discussion to talk about this. I mean, there's ways that we could talk about that which would be much more illustrative. But to me, that capture, and we should do those at some venue or other probably, because I'm sure this doesn't make sense to a lot of people. But that is the most immediate kind of connection around that sort of thing that I've seen recently.
So if I can just jump in for a sec, it's Jennifer. I am a little bit worried that we're going into kind of a big, broad discussion about modeling here, which is, you know, like near and dear to my heart for sure, right?
I was hoping you'd speak up.
modeling programs that we all know well, but I'm wondering, you know, if and you know, you guys have brought up lots of really good ideas, and I think everybody's sort of preaching to the choir here with the biological questions driving models and focusing the models and lots of different varieties of models. And but this is a bit diffuse, and I'm wondering, you know, if there's some way that we can focus it back to the, you know, cross-atlas integration or within atlas integration, or you know, kind of a little bit more where you started, something slightly more tangible than this large discussion space.
Yeah, no, thanks. I think that's helpful. And I think it's helpful to hear from everybody about what the challenges they really see, because I see part of our opportunity here as outlining the challenges so we can work more effectively. And with these great Atlas initiatives that are generating heaps and heaps of also new data types, how do we start to, you know, either reformulate the questions or actually be able to address some of these biological questions given the new data types, and maybe that necessitates new models as well. So I think.
Could I make a suggestion? Yesterday we had sort of a similar, in the session I was moderating, we've got, you know, so many things to talk about, where we really ended up focusing was if we had just to find one specific problem, maybe this is even an opportunity to break out further, but to sort of weigh in on, from all our perspectives, how we might solve that problem together. And then at least that's sort of an actionable thing that we could take forward, you know, that, you know, maybe bridging across atlases. I'm not familiar with all the atlases and where the bridging opportunities are. So maybe that's even, you know, sort of a first step, a first question, you know, where are the bridging opportunities? Just suggestion.
Great. No, I like that. Peter, I'm going to call back on you because if Peter's still here, there you are. I think you had some concrete examples, and I guess I'm wondering if, especially where you're sitting now with some of the H10 efforts and the imaging data, but really thinking about that in the context of the kinds of systems pharmacology, because I think that bridges 2 worlds. I mean, is there a question that you see being formulated from some of this that's central to these Atlas topics that we could sort of bite off and chew on as a group here?
Well, you know, the most obvious one that came up when we were also thinking about a session we're supposed to run this afternoon. So often these different sessions are coming around to the same questions. You know, these atlases are going to have, many of them are going to have data really on cell states as defined, you know, are cells dividing, are they next to, so they'll often come from a method like imaging, for example, are they dividing, are they, you know, what is their morphologies, et cetera. And then they're going to have molecular data, most simply and most probably initially single cell sequencing data. And so, you know, how those things get mapped from single cell sequencing back to cell state is, I think, one of the questions. And the place where we see success is in something like immunophenotypes, right, where there are fairly well-defined marker spaces. But even that, as you probably know, is quite tricky, because things that show up as dominant markers, let's say in immune phenotyping in flow cytometry, which has been classically, you know, CD4, CD8, are not actually dominant features in gene space. So there's already a fairly subtle mapping between a purely data-driven definition of immune cell types and then classic markers. And I think, you know, that plays out all the way through. So that's what we've been often talking about. And give you an example, in tumors, you'll often have these so-called dedifferentiated states that are thought to be a mechanism of drug resistance. And they're defined by a set of markers in gene space and a essentially non-overlapping set of markers in protein space. And the relationship between them is largely unknown. And it could be tackled with a set of experiments, but those fall outside of the atlas efforts in many cases. But I think one last thing just to say, I think Fiona had an interesting point, which is I actually think the activities of many of these atlases are not so obvious. And in the previous calls I've been many times, on this meeting, the last two sessions, people say, oh, we have this, that, next thing, but oh, it's in press or it's about to get submitted. So I'm surprised at how relatively little use there seems to be of preprint services in all of this, right? So you know, we've been trying to use preprints for everything in the atlas so that you can at least see it. So maybe there'll be more visibility, but the ability of atlases to look at each other, if it's relying on published papers, is going to be, you know, 2 to 3 years out of date. And so I wonder if there's a way, this poor Jennifer, I'm going off on a tangent again, but I wonder if one of the ways of actually achieving some of this integration, a practical thing, is to see what people feel the barriers are to using preprint servers or to pre-publication sharing. Now, all the NIH efforts, we've all committed to pre-publication sharing. It's just not actually happening.
Hey, this is Jonathan. I can make just a quick comment on that for HubMap, which is supported by NIH, but it has its own PIs that, you know, drive policies and things like that. Across the consortium, we've committed to a sharing policy that requires you to send to a preprint server at the time you send for submission for review. So I think that the landscape on that is changing pretty rapidly, Peter, and I'm not so worried about it anymore. Okay, that's great. HTAN has done the same thing, so maybe we're looking a little bit in the rearview mirror here. Yeah. But I do think that I think one thing I noticed in HTAN, and this gets to data as much as it does to get to models, and that could be concrete, is I do think despite the fact all these efforts have data coordinating centers, the effort to get the information out relatively efficiently and to record what the activities of the various programs are there's not much of that. So it's actually hard to go from one effort to the next and find out what they're doing. Yeah, I think that you point out is a remaining challenge. It's particularly a challenge for us right now in HubMap as we're under pressure to get things engineered and developed to get first data releases out, and you know, get the documentation and the GitHub repositories in the right kind of shape and all this kind of stuff all at the same time, so that when it comes out, you can have communities that have little knowledge of it that can approach you, communities that have very deep knowledge, communities that want to use your software, use your data. I mean, there's so many audiences to simultaneously deliver for in these DCCs that I agree we spend the time on the engineering and getting the basic functions out, and some of these communication parts of it are lacking.
One possibility to be concrete here, although I don't want to actually propose a solution because that would seem like we were doing what we were supposed to do. But one thing, I'm wondering if there's a possibility, I mean, one way to get data across diverse communities that Jonathan points out is typically that needs curator. It needs somebody to do a curation function, right? And if there's something we ignore much more than models, it's curation. But anyway, I'm wondering if it would be possible for all of these various efforts to it could be a fairly modest number of sort of data model method curators that would have access to everything. I think it speaks to Jonathan's point. I know for HTAN, it's the same thing. It's a full-scale rush to get that first data release. So this is not on anyone's critical path. But you could imagine a meeting like this if there was again, a fairly modest, it could be even 2 or 3 people who across all of them were helping to curate, where are things, what are roadmaps. I think you would see, it would be much easier to find resources, and those people would be navigators. So almost all programs have that internally, but we don't have that across programs.
So something that Peter brought up quite a few minutes ago was about self types versus cell states.
Mm-hmm.
And I actually think this is really important when you're looking at things like the Human Cell Atlas. Is, I could say a B cell is a cell type and a T cell is a cell type, but actually, I had this model this morning with one of my students, and the T effector memory cell, is that a state or is it a cell type? And actually, these have become very important as we get more and more genetic information. Is really, we all accept a proliferation as a state the cell's in. But actually, some of these other things, I think, are not really well defined, and this becomes very important later on in terms of the modeling.
Yep.
Yeah,
I mean, I think that's actually an excellent point, and I wonder if I'm hearing that as something that maybe as a group we could tangibly, think about ways to curate, what the curation, what are the approaches that are necessary for that. And Anna, you had something to say.
No, I was just saying about the curation of the different database and consortium. I think is a great idea, because you have to know what they are working on, and you should know in a consistent way between them in order to go back and forth and query between, know what they're doing and what we have. And so I think that is a great idea to propose something like, curation between them. And the other thing I was respond to Mark about the cell and cell state. And I think that is a very, another interesting point where, for example, from the ontology point of view, it's not very clear how to respond. And I'm an immunologist by background. And I know that, really is very complicated from the immune cells figure it out when you are really defined as a clear T cell, or in a state where a T cell is for a certain period of time doing something, like upregulated gene, produce cytokine or do whatever, and then go back to what was before, or if it's really a different type of cell. And I know that is one critical point. And it's not, I can tell you that from the ontology point of view, the states are something that the ontology looks as weird, because it's not very easy to define states. And so I think that could be, but I'm sure that is something that is needed, especially for the immune response or any kind that involve immune cells.
I wonder if it's possible to almost imagine these two being one thing. I like very much Mark's picking out the issue of curating cell state. It's related to cell type, of course, in some ways, and cell type is definitely something the HCA is working hard on. So you could even imagine a cross-cutting kind of effort of this type if we thought that had some merit, but that actually would, as an initial work product, looking at cell state, right? Now we have proliferative or not, EMT or not, and then we give up. And it would be, I think inflammatory is another. The problem with inflammatory is it's, a lot of different things. So getting a more subtle definition of those would be an interesting piece of science, and would, I think, be helpful for this modeling. But I think it also might be tackled across all the efforts. Somebody would have to take it on it as a discrete thing, but you could potentially kill 2 birds with 1 stone.
Yeah, it would be even important in trying to define the cell that comes from looking at the single cell RNA-seq, because there we are trying to define the cell as a cell type, but sometime in the middle, you could have different states of the same cell type.
Right, and to what extent can you leverage these perhaps additional data types to do that? I mean, right, so we sometimes think about whether we can define those through some of the like site-seq based assays where you're coupling the RNA to some immunophenotyping, or you're looking at an accessibility state. So attack-seq type methods to try and flag whether this is in an active state or not. And of course, this whole field has evolved so rapidly, much driven by what's going on in the immunology space. But I do think that would be something that perhaps there is a group that cuts across atlases that could tackle, and that's something tangible that would, I imagine, be of great value to the whole community. I know there's special.
Christina, I wonder if if we wanted to focus it even further, just as a first thing, actually just defining Mark might be able to comment, just the concept of an inflammatory state, right? We use that word, but it's very different in different disease settings. We sort of have sterile and non-sterile. We have a couple of common subdivisions, but to actually get some molecular definition of that would be pretty interesting, because the gene sets, if you go into geo, that define an inflamed cell state in different tissues, they're barely overlapping, except you'll pick up interferon in half of them. So even just a better cell state definition around something like that that's really coming into focus would be interesting, perhaps.
Hi, this is Yan Su from LearnMap and the Learn Network seats. And I agree, many of you talking about the cell states. And we started the learn development and also disease. You can see we identify a bunch of different states, right? And some of that is a deviation from the normal states. So it's almost impossible to clear define, because it's a dynamic. It's change the process. So basically, I think all the consortiums has their own role so it's actually pretty urgent to define a clear guideline, so in our case, we're thinking unless this states is like during the development, it's actually, leading to a clear branch to another states or to another, definitive differentiation type. And you can observe in the morphology, in the immunofluorescent states or something, followed by marker, and then in the cancer stage, you can see it's deviated to a totally different cell, and then we define that as a disease state. Otherwise, it's probably not necessary because it's a continuation differentiation, so it's hard to catch. I think it just define the cut point at I guess different disease and the different organ will have the different guideline, but the guideline should be clear. Then all the consortium can be consistent using that.
The one field, I mean, we could talk about inflammation or fibrosis for a long time. The one field that we haven't captured intended to be is pathology. There is nothing like the eyes of a pathologist. They look at stuff and they see stuff that I can't quite understand how they see it. But I think it's not really capturing state, but there are features, I would use it that way, which they have learned as a super machine learning human, which I think we do need to capture somehow. But that was just a thought that's in my brain.
Mm-hmm.
That's a great point, Mark. That came up in our meeting yesterday as well, around the concept of having an anchoring point, and perhaps the histology pathology is, at least at a macro scale, the sort of best anchoring point to then scale up or scale down from there.
I think that has also emerged in HTAN. I'm sorry, Christina.
Go ahead.
Has emerged in HTAN. There's actually a fairly large number of pathologists, because it's cancer, but they've been what we've observed is they've been very much separated from the molecular work in HTAN. So one of the linkings is going to be via imaging, and in particular getting the H&E imaging that they're familiar with respect to Mark's point about cell state linked to these molecular images. I don't think it's a technical challenge, but it hasn't happened. And as we all know, digital pathology is just coming in very, very quickly across the globe, but it hasn't been very linked for the most part to the activities of these atlases.
Yeah, I was just going to add to that, Peter, that's something that this kind of helps close the gap. And I see this distinct challenge of defining cellular states when we have dynamics. But I think the topography and the tissue landscapes, you know, whether we're trying to understand this in the context of the topography of cells within, also, you know, in relation to vasculature, I mean, that's a different layer of mapping, but one that I think, at least in my view, helps to ultimately bridge these multi-scale models. And I think, you know, we've identified this rich challenge of mapping and identifying cellular states and particular classes of states, I think is what I've heard. Those of you working on that probably have a more elegant phrasing. But I would love to see this be built in. And I guess a question that I would have is, we've heard from different people in HTAN, and we've heard from Fiona and just now from you from the Long Map Consortium. I mean, is there a way across these consortia to do that? And are there people that, you know, what are the other missing flavors of data or information? And do we need the data coordinating centers talking to each other to do this? I mean, how do we get investigators on an individual level to annotate, right? I mean, we need to be able to
I'm going to say the same thing over and over again. It's the same thing by mistake, but just to emphasize that, sorry, is actually, I think this thing with, you know, getting pathologists closely involved in this proposed cell state annotation across the effort. I don't see any of them having substantially engaged. We'd have to think how this worked practically. But what is not named cannot easily be studied. So, you know, one of the things we've noticed in our lab, where we're doing a fair amount of imaging on both lung and melanoma, just to give you two extremes. One thing that's interesting in fairly advanced lung cancer is there's an enormous amount of pathological annotation on feature sets, which means you can say, that is a such and such feature. What is its molecular attribute? It's interesting, as you get to advanced melanoma, you and I would look at them, and these are wildly different looking tumors in molecular space and visually, and people go, that's just metastatic melanoma. So it's never actually been subtyped and named, and that actually makes it incredibly difficult to now study it from a molecular standpoint. So this process of naming things and having features against which you can now bring a molecular thing is absolutely critical. And I think it is a restatement of this health cells type.
So I'm a pathologist, and I'm currently working on single cell data. I raised this point at our spatial profiling session yesterday, that I feel like a pathologist is critically needed in especially the spatial data aspect of the cell atlas, because we can help in terms of, you know, delineating morphology, adding an extra dimension to the data.
Yeah, I agree. And I'm working in this project where we're trying to make automatic recognition of particularly cell type in histopathological images. And from my side, what I'm trying to do is try to represent formally the tumor this is in tumor, so the tumor microenvironment, trying to represent what is the tumor margin, you know, intratumor, all these spatial relation between different type of cells. And that's is something that I think the model and the computational people, because I'm not that, they are going to use in order to then build their model. And that we have done this in the lung map phase 1 part, in using it, looking exactly at these were immunofluorescence images, and in collaboration with the pathologist, we figured out which marker we're using to distinguish the specific cell, and then build a small ontology coming from the large ontology. So I didn't really build up any new terms, but just grabbing term and make it this representation of what the pathologist really does. So a little piece of what is in mind of the pathology, of the knowledge that is in the pathology.
They're very familiar with the markers.
Because obviously we cannot, will take time and effort that we didn't have it. But to build the knowledge that the pathology has. So what the pathology does when you look at the images, how it can discriminate between, say this is not that. And that was an effort we have just sent in the paper that will be published, where you have, for example, the ontology representation, the knowledge You know, it's kind of little piece of the pathologist's brain to drive the algorithm to automatic segment the images. So that was one approach. And I think that's, you know, something that we should kind of pursue a little bit more, but it needs to be, and this work was a collaboration with pathologists, computational ontologists, and biologists all together, because I think that's the key, you know.
Pei-Ling, do you think it would be useful to, for groups like this that are fairly research focused, to reach out to, say, CAP,
Yes.
the College of American Pathologists, or the Royal College of Pathologists?
For example, in my group, we have pathologists. I actually do single cell work. I'm interested in research. And I think targeting pathologists at each, you know, institution, or I don't think a whole lot of pathologists are currently involved in single cell effort, or at least a single cell ADLA effort. And I feel like it's a total must, especially if you're going into the spatial domain. I think single cell RNA-seq, you may not need, like, you know, the architecture, the structure, or landmark as much as spatial data.
It's interesting, you know, what we've noticed is there's not been a lot of crosstalk between research applications, and you would see that, and the rapid emergence of digital pathology in general. We actually wrote a paper about this recently that just popped up on bioRxiv, and it bounced from journal to journal because all the clinical journals said it was far too much research, and all the research journals said it was far too clinical. So, you know, there seems like an obvious, powerful crosscut, totally related to the rest of the discussion, that just has reached this kind of social barrier. There are clinical journals and there are research journals,
Yes.
and something that tries to span the two is in the famous Valley of Death.
I've seen that too, Peter, but I guess, and I think this is going to be a challenge going forward with a lot of the HTAN efforts. I wanted to touch a little bit on two things that might link that, which is to the point that Anna Maria had made. And it sounds like you've already made a lot of good progress in trying to, you know, work collaboratively to define these cell types, something that has been sort of a wish for many years, and I think we might have the data to start to do, is precisely this: to take these annotations, to understand the types of cells, the numbers of cells, so population sizes and their topography. And what we're really keen to do with that is then to bring that information in to these agent-based models. Sorry, I went off on this tangent before. I know some of you have worked with these, Mark. We're trying to really, you know, we felt very much that so far these models have been very naive in that they're mostly tumor-centric because that's what we know how to model. Let's start with the colon, where we have a crypt and it divides, and we can track cell division up the crypts, and maybe we can even computationally simplify the model because the crypt splits in two. Let's assume it divides by fission. Where we've really struggled and kind of hesitated to do much because we did not know how to parameterize the models. And I think if we're going to talk about models, you know, parameterization is key. But we really are hopeful that with appropriate annotation by skilled pathologists that know what they're looking at across these tissues, that we can start to build that into these models. And you know, one of the challenges that brings is, well, how generic of a model is that? If every tissue, every patient has a different composition, you know, how variable is that going to be? But what we're trying to learn at a baseline is what are the types of cells that need to be incorporated and their numeration. And if we had one tissue type to work on for that, we could start building these models. It's taken my lab a long time to get going on the imaging, and we've been doing this through HTAN, through other efforts with Codex, with MIBI, and with NanoString. But you know, we're heavy on the computational side and trying to bring this in. And I think this is something that there is a real opportunity to actually start to bridge scales here, but it takes very, you know, all this expertise that we've talked about. And I, for one, would be really interested in engaging with others that are interested in, you know, thinking about how we could get the measurements. And I know that's something that Peter's been doing a ton of for a very long time as well. And then build them into these models. And the other component to that, you know, if we want to get down in the more granular, has to do with segmentation. And we too have been thinking about ways using additional marker sets, so cell membrane markers, to improve segmentation. And there's folks working on that for methods that we think will be deployable, hopefully available this summer, for all types of immunofluorescent, so cyclic IF, but also MIBI types of data. And, you know, that's one component, but I think that's a small component of this whole picture. But but if there was interest in this group, I mean, I think it sounds like there's a lot of diverse expertise here that could help think about, is this a problem that could be tackled on some level?
Well, I still think, I was just come back to this thing. I wonder if we could have, if the list has whether somehow at a consortium level or a consortia of consortia levels reaching out to these pathology colleges, right? Because
Yes.
they have whole working groups dealing with digital histology. And what's happening in digital histology is moving from the glass slide, obviously, to scanned images. They're H&E, and then very heavily being influenced now by machine vision systems and deep learning in order to get phenotypes. So they all have working groups. I'm sure Pei-Ling knows much more about this. And they largely disconnected from the research, much more so than even research. Now you're going to correct me, but I get the sense in radiology, they become a little more connected.
So I think there's a subset of pathologists who are more interested in what we call basic science / translational research than pathologists who just practice, clinical, sign-out cases. I think you may have to target the group who have more interest in this type of research pathology. Like for example, for myself, I'm working on single cell data on skin cancer. And I actually have never heard of this. I heard about this human cell effort, but I wasn't sure how to participate. So I basically signed up on your website and, I saw, oh, wow, there's this meeting. And I came to see what this meeting is about. And yesterday at spatial profiling, I embraced the issue that I feel like you need a pathologist on at least the spatial profiling effort. And I think maybe some pathologists just don't know how to contribute to this.
Yes, I think what we found a little bit, Pei-Ling, I don't know if you can comment, is that in our institutions in Boston, we have, like yourself, pathologists who really, are translational research scientists, absolutely, and they're key. But then there is this rather larger community of practicing anatomic pathologists who could help with some of what Mark was talking about, this sort of cell state definition, and they're now being dragged into the digital world. So it's to see how many of those, and we've been surprised, 1 in like 5 of them, generally the younger ones, tend to be actually pretty excited by, in the same way that oncologists are excited by genetic data. These people are like, oh my God, my field will suddenly, be recognized for the fact it makes 95% of all diagnoses. Anyway.
Yeah.
Well, there's also some
Send me an
strong
email.
groups in Stony Brook. You've got Joe Saltz there. There are already some real front runners in the digital pathology space. The TGA pathology database. There was a really nice publication a couple of years ago on the machine learning analysis of those images. And so I think there's already pockets of pathologists out there, like Pei-Ling has said. We've been working with pathologists for years too, and it is sort of a hard transition for sure to go from the spatial or H&E to the spatial analysis of tissue and believing that data. There's like sort of a constant battle over the last 10 years of comparing fluorescence data back to brown stains to ensure that the staining, because you lose that amplification as well with fluorescence. So, but I think, we just need to get more pathologists engaged. And I mean, there's plenty of interest. I think to your point, Pei-Ling, you didn't know about this group. So, maybe even a deliverable from this session would be a working group paper, maybe encompassing all the sessions, but to sort of get it out there and what the whole scope of all these different atlases are doing.
In LabMap phase 1, we actually, have a ontology committee led by 2 pathologists, and so we have a complete hierarchical structure of the tree of the length from different development stage, and we incorporate into the single cell data into the new cell types, all that. And phase 2, we're moving into the disease stage. And then again, the pathologist plays a really critical role. And they actually, for the disease, they define the regions and pick they're the first line actually, pick the, similar region to compare with the next, around surrounding regions, all that. And in terms of modeling, I agree with Christine. So I think the parameter adjustment probably is most important is first probably is dissect based on organs or based on tissue. For example, lung, and then you modify the parameters, adding different cell types to the different organs.
Mm-hmm. Yeah, I agree. And it's great to, I mean, it's great to hear that folks are engaged in that. I think within some of the HTAN efforts, certainly some of these projects are being led by pathologists. So on the breast front, Rob West is heavily involved with a lot of the, both the imaging approaches, but also technology, single cell data types. And I know there's a handful, but I think, you know, it's also important if we're going to really have this curation across states, we need to be talking to each other and not developing our cell state annotations in a vacuum, because that is sure to lead to problems downstream of that. So I guess we have a half hour left before we are meant to break. And I think we've had a really robust and exciting discussion. I'm very, you know, excited about a lot of the topics that have come out of this. I can see people are as well. And I think it would be good if we can think about what are the concrete items that we might actually be able to follow up on and see through from this. I heard a lot of discussion about the cell state annotation and curation, and how we could do that across atlases. So maybe that's one point we can drill into further. And I also heard about bringing in, you know, and I think tied into that is the topographical, the pathological annotation, and I guess how we can define the cell states to some extent in context of these tissue architectures. Is that, I mean, is that something that we're really going to have to go from single cell dissociative data in some respects down to mapping into tissue? I don't know, that one's a harder one to tackle, but.
It's suddenly moving into complex ecosystems.
Mm-hmm.
And it's always difficult to know quite how to embrace that, but how to capture that. Because each tissue is its own complicated ecosystems, and tumors develop their own and change them over time. But capturing that, yeah, it's just something at least to think about.
I agree. So divided by organs or tissues is the first step. For example, a lot of consortiums doing lung or doing kidney, all those consortiums should be meet together, you know, see the guideline, unify the guidelines.
You know, I think the fact that we heard from Pei-Ling and others, and that there actually is, you know, is this crosstalk with pathologists. Just to come back to that topic for the moment, you know, the fact is, I think our center has 8 or 10 of them involved. I think that actually is a huge strength. I think, in other words, I think that's good. If there was no such discussion, we wouldn't have anything to build on, right? So I think that, you know, I think all the action items coming about out this are related but separate. I think one of them is this curation of cell state. I think that could come from many areas. I think the idea that we actually need to dedicate some effort to cross-atlas annotation and learning. It's just not going to happen. It'll happen naturally as papers come out and we read them, but that's fairly late in the process. So that actually would need to be resourced. And then the idea that we would reach out more to anatomic pathology, which is particularly in the tissue space, has a lot of knowledge that I think could be gained. You know, all of those could come together, but they're also somewhat distinct and could have individual work products associated with them. And I do think that the emerging area of, as I've said before, the practical application of digital methods in diagnosis and pathology could be more tightly linked with our new research areas on these atlases. One thing we did, we were actually contacted by the FDA to help them think about how to score the reliability of machine learning applications that are now being used to diagnose H&E. And so they put us in touch with, there's some academic effort, there are 4 or 5 major companies doing this. And we said, okay, well, why don't you recognize, you know, B and T cells, tell us what your algorithm does, and we'll now score that using a molecular method. And it was pretty striking. No companies were interested in that, right? Because I think what you would find is that those algorithms are still quite problematic. And so I think that's the kind of thing that has to happen in the public domain.
Mm. Yeah, that's a great point. I mean, I think to just on a high level to think about linkage back to these integrations. I mean, digital pathology is this whole field unto its own right that's rapidly emerged, but I think there's some really interesting opportunities as well to think, as we have these mappings, how can we impute information that comes from, let's say, bulk gene expression onto these digital pathology based protein readouts, as an example. And there have been some efforts to do that. Similarly, there's been some efforts to apply sort of deep learning approaches to take the H&Es and try and infer somatic mutation state or copy number. And these are all pretty new and very much in development, but I guess I would just highlight that I think there's an opportunity for that to be a platform for how to integrate some of these different data modalities. And we talked a little bit about that earlier as well, which is, to Fiona's questions around modeling apoptosis, right? And how to link RNA back onto what we're reading out at a protein level. So I think this, there's some concrete things that need to be done to make it feasible to actually model these data, but there's a lot of opportunities to have that.
Christina, one additional work product, if you thought it, or the group thought it was appropriate, could actually be in the area of sort of education. We started off this particular meeting by all giving these little webinars. And if you actually make your way through them, there's a huge amount of interesting material. I mean, it would need another pass through to make it, at the quality of, what we try to do in a flipped classroom. But for example, again, in the FDA, I think the point you brought up earlier about machine learning is you want to have the best possible data. But when the data is challenged, machine learning can actually extract features from the totality of the data in the face of considerable noise, right? I mean, these are sort of well understood or becoming well understood in your discipline, but I don't think they're more widely understood. So I think also another aspect of this in general could be to have little working groups produce more polished lectures, even if we were lecturing to ourselves. Because actually, as I went through the prep for this meeting, I was looking at the videos and I thought, oh, that's an interesting point. I never thought of that. So I think it's another way to build cross project collaborations, is actually to collaborate together on producing some of those pedagogical materials. That's how we all end up collaborating in universities, is we have to teach the same undergraduate biochemistry course. So it would build on that.
Yeah, that's a good point. And I think there was a lot of, certainly I feel like I would have a lot of learning to do to understand the nuances of a lot of these different components. And I suspect we all feel that way. We come from our own domains, and we're really talking about quite multidisciplinary efforts, bridging from pathology to these sophisticated technologies and then analytics to interpret them. And I would separate the analytics to interpret the primary data from the models that then integrate them. And we already heard a lot, had a very healthy discussion about what kind of models are we talking about here, and data structures and so forth. So I think there is a lot to be learned from one another on that, and that certainly could be an output. I know there's some crosstalk between each of these, between some of the sessions that have gone on yesterday with the spatial.
I mean, it also makes the observation that, the most robust social systems are learning systems, right? I think that, and that people teach each other not because they're experts, but because they need to learn the topic, right? So the very process of teaching is the way you learn. So anyway, I wouldn't want to overbear it, but I think this idea of building this that we've been talking about, cross annotation research, may be focused on cell types. It could also have a kind of pedagogical aspect to itself that actually would facilitate rather than interfere, I think.
Right.
Christy, I have a question regarding to the modeling, you're talking about integrated protein and RNAs, and we know it's poorly correlated. In terms of a model, should we do separate model? Maybe it's even confusing, right? Because we don't know the underlying mechanism. Some of them why it's correlated, most of them not correlated. At this stage, what are you guys doing? Are putting together or actually separately?
It depends a lot on the biological question. We build various different classes of models. I think the one most relevant to what we've talked about here is we've done some we have data sets where we've looked in tumor atlases through time, so pre-treatment, on-treatment, post-treatment, where we have bulk gene expression, and then we have done spatial proteomics, and then we have some spatial transcriptomics, maybe more limited. And we've done quite a bit of pathology-based assessment of, in this case, stromal TILs and infiltrating TILs, and have really just been trying to understand. I mean, honestly, our first view was, what do these data tell us about this process that really hasn't been studied? What I will say from that is that we learned very different things from the protein versus RNA models. Perhaps not surprising, treating with a targeted therapeutic agent, we were expecting to perturb the protein network, and we saw that and found that we can maybe predict pathologic complete response pretty early on, whereas the RNA was far less informative in this regard. And so then, you know, looking at those in independent models, I should say, you know, multiple protein features, both tumor and immune, then it was kind of clear that actually integrating it, we didn't get a huge amount of benefit from the RNA. So that was a model that's more related to this digital pathology type question we're talking about here. I will say in other contexts, depending on the biological question, I think there's huge value in integrating it. And I think what we're trying to do is learn, in some cases, relatively sparse signals, right, that are attenuated maybe in this bulk population because you have many cell types present, and we're looking at a mixture of cellular states and cell types. So I think it depends on the question. I think that deconvolution is a hard problem. And so it depends, you know, it depends on what the question is. I'm not tossing the baby out with the bathwater. We certainly use that for other biomarker related questions. But I guess, you know, again, there's also a challenge of what are the measurements we've made. If we had matched RNA and protein, we could really try and evaluate that. We would also want phosphoprotein. You know, and this maybe comes back to some of the other questions around, you have to typically design a multiplex panel for a lot of these assays, and, you know, what's been included there.
Christina, do you, I mean, and this came up in some of the other groups, so it might be covered elsewhere, but just given the diversity of measurements that are being generated and that you clearly need for your models, do you have challenges in the preservation of your methods, and of your samples, and how are you kind of addressing that to sort of ensure that the correlations that do exist between a protein level and gene level and so forth are actually maintained? Because of course the time to fixation is going to be a huge variant on that.
Yeah, that's a huge challenge and issue. I think we even have a working group, and we certainly do within the Human Tumor Atlas networks, for how sample types are being stored and then ported across methods. I think there might be even a working group from this meeting, this virtual meeting, that was tackling some of those issues. I didn't have a chance to join it, but we do. And I think there's real challenges. People are thinking about preservation of nuclei and then doing the single cell RNA and attack measurements. And, you know, obviously how we process samples is a big consideration. That did come up actually yesterday in a multiplex. I think you were on that call with, actually, we were on the same one. So, you know, I think that there's some discussion of that. It's hard if you want all flavors of data. And I'm not sure, I mean, I think a group does need to address those biases. There's also just the practical considerations that do differ by tissue type. I guess, yeah. So I'm not sure if that's something that this group can really address, but I think is clearly.
Yeah, no, maybe not. I'm just, yeah, I know it'll probably be taken care of elsewhere, but just in terms of thinking of you know, the sort of the ideal gold standard data set, where, you know, sort of bringing it back to what the original, I guess, the overall goals here in this group. But it seems like that would have to, you know, if you're going to do this as a sort of cross atlas effort, then that would have to be a sort of a prerequisite for sharing of data, that and in order to bridge these different scales. Yeah. So maybe guidelines. We talked about guidelines yesterday. You know, that seems to be probably going to come out in some shape or form from this. And I know it is happening within each of the atlases, but there have to be consensus across the groups in terms of how, you know, samples are preserved.
Yeah, it's a great point. I mean, I think it fits in with almost, well, to some extent, this curation component that we do need to know what we're working with. And I think even to the point that I believe Ian made earlier on, you know, pulling down data from this data portal where he found organoids but wasn't sure that they were matched. I mean, just as we need to know the tissue of origin, we need to know how samples were preserved if we're going to compute on them. So I'm looking at the clock and we have 15 minutes left, and we've had this really, really wonderful discussion. So for those of you that are looking at the Google Doc, our task in the next few minutes to make sure that we all remember what came out of this before we go on to the next series of topics is we get to give our group this collaborative discussion a title, and then we can talk about some of the recommendations, problems that we think that we can uniquely solve, and folks that would like to be involved. And a thought, but I would like others to really weigh in on this is, do we think this is just about cross-atlas cell state curation? Do we think we can get into topographical mapping? Does somebody have a catchy title for what we're trying to do so that we can revisit this?
And can I just really quick note, so currently there's 3 blue boxes. There's one that's focused mostly on the state curation. Then I added a second one for the engaging the pathology community. And then there's a third one that's much less fleshed out on the education outreach stuff that that Peter mentioned.
Got it.
I was currently separating them based on Peter's early comment that sometimes we keep the problems too big that they are hard to eventually solve. But if it makes more sense to keep the pathology and the cell state curation together, then that's fine as well.
I think it's great that we have them there and we can go back. I mean, I agree. We want to have tractable problems, not that people come back and think, oh, we can't make a dent in this. I think we can make a dent in it as a collective. And I think this is, you know, really cuts across all these atlases. So thank you for doing that, Sean. That's immensely helpful that you've been taking such great notes here.
Should we give some suggestions? I know it's not a unified, the answer, suggestions on a model, you know, like the top potential models can be used for the integration, like
yes,
regression based, you know, deep learning based, something like that. People probably looking forward to see, you know.
Yes, we can probably bring that down here. I think, guess it depends on the biological question, but I think these methods come into play in different places depending on the types of integration.
Sorry, I'm just going to try and capture that. Is that a question? Is that a problem that we're going to solve? Is the curation of the different methods used?
I just say maybe it will be benefited to the community in large.
Okay.
So right now, just if we look at the first kind of, I think, cross-atlas cell state curation, that was one of the themes. So having a group that's focused on developing these approaches for curating cell states. We named some of the states. I guess ultimately we may want to decide how many of these we think we could tackle. Mark and others and Peter and others had suggestions around inflammatory cell states. We talked about you know, EMT or proliferative as well. I guess just to drill into this a little bit further, what do we think we could bite off and chew on as a group, you know, across these atlases to define states? We want multiple states in there, I assume.
I guess one of the things is we just need to make sure we're listening across communities. Immunologists have their own view of the universe, which is often different from somebody who comes from the developmental biology field or neurobiology. And we all come with our field biases, but it's also important that we capture aspects of fields. Otherwise, people feel like they don't want to engage because they feel like it's described in a way that is foreign to them. Immunologists love to put everything to CED antigens just to annoy everybody who's not an immunologist. But I think some of these, that's just a general point as an immunologist, because I say CD this, CD that. But I think we need to pick up some of these field things to make sure that in the future there is a really good buy-in.
Yeah, and I think that's thinking about which, if it's better to go for, specific type of cell state between, or, the general cell state. I think we have to start from the single cases, because the cell state is, I think, is really related to the context. And in particular, what was saying from the immunology point of view, what you can define inflammatory is a mix of things that could depend, on where you are and what is the balance between, for example, if you want to say which are the inflammatory cytokines, and Mark can correct me, is not depending only on what you are doing it, but which is the balance between them. So between the anti-inflammatory, the inflammatory, you cannot just be sure that if you define as an anti-inflammatory, then you will have the anti-inflammatory environment. So I think it's very related to the context, what you're working on. And that's why I think the cell state could go from the specific context, and then, taking the same time of approach to define that should be the common approach. And then maybe from the specific one, we could, if put all together, we could come out with something that is more general. That is my opinion.
I think this, I think to Anna's point, I suspect that actually, as you try to define cell states, absolutely in terms of tissue context, but also in complex settings, they're going to also be geographically different. I think what you're going to see is those cell states that have the same name today are actually very different from each other, and conversely, which is exactly what's happening in single cell, as single cells get defined by sequence space. Things we thought of as being quite different are actually quite proximate, and things that were thought to be, oh, these are very closely related, are very, very distinct. So, I mean, inflammation, fibrosis was another one that Mark mentioned. I mean, these states are defined, but they have this name, often coming from maybe even a pathological thing, but I think they're very different in molecular terms. So cell state definition is absolutely a contextual thing. And then the interesting part will be to see how similar and different they are across contexts.
Mm-hmm. I agree. I think those are bold but tangible things that we could work on across atlases. I guess Anna and Peter, you've been very vocal.
Do those have agency?
You have agency.
Okay.
I think if you're happy, we would like to put your names down here. Whoever would like to, be engaged on this. If you want your name removed, I guess we can find ways to do that too. But I think Anna and Peter, if you're happy to sort of be listed as affiliated with this cell state mapping, and Mark, I don't know about you, Fiona, or if you gravitate towards any of these, but I think it would be nice if folks that are participating in this are willing to continue the dialogue and see where we can go with this. I think it would be really, really hugely valuable for many atlases, and hopefully we can engage others on the way. I completely agree that the context is important, and I guess I see this as the cell state mapping as really jiving with putting it in the context of the tissue itself, so with the spatial approaches as well.
Christina, just I think, right, with regards to the fact that I think this would be relevant to many atlases, Toby sent a message there in the chat to everybody asking if we'd be willing to have this as one of the soapbox presentations at the closing.
Oh, maybe we lost you, Sean.
Enthusiasm about it, to ask the community to get involved, if this would be something worth suggesting for that.
I think it would be great. Would anyone on this call like to lead that? It's 2 minutes, no questions, soapbox thing. So pretty brief, no slides required. If anybody.
I vote that you should lead this, Christina. You're leading our group. So I think you would articulate this well and effectively.
All right, I will do that for the group. Are we happy seeing this combined? I think this was to Sean's point earlier. We had these sort of 3 breakout topics. This, I think, critically integrates the pathology and the annotation across sites. So I'm happy to see those combined. Sean, you were asking about that. in my view, unless somebody else thinks that's too much of a reach. This was the efforts to engage pathology and define these, but I think we have to be defining.
I think it makes sense to incorporate it in here because this then addresses the question of multi-scale and then bringing it back from histology to cell state and onwards. So I think it's a good way to bridge the two as well as bridge the biology.
Great, I agree with that. And I think if there's other concrete, so everybody can edit this. And if you have not been editing this document and you want to, or just even want to scroll down to the blue box, we'll go back and parse it. And then for those of you that have seen the other ones that came out from yesterday, pretty nice concrete just output, so that I think it will help others if they want to go and view the video or they want to know what came out of this, what we talked about. So please do look at it. You guys all had a lot of great thoughts, and it's hard to make sure that everything was captured. And even if it wasn't discussed and it should be discussed in the future, put it in there, typos allowed. Doesn't have to be beautiful or anything. I think that'd be great. And then Peter, I guess I want to come back to you a little bit. You had the suggestion about ways that we could educate each other. And so I guess a question that I have, and maybe folks involved in the atlases will have a thought on this. We had these very brief introductory calls. Peter, you and I did one of these for the HTAN, right? So where we talked about the atlas at a high level. Are you envisioning that an output of this series of discussions would be short talk that really gets more into the technical details of what people are doing? And is that something we want to put out there and see if it gains traction?
Yeah, I was trying to think of something that the reason I suggested it was in part to come up with something completely concrete that could happen, right? So I think if you're going to do cross-platform linking, you're going to need somebody who does it, right? Everybody's going to have to pitch together, and whoever the funders are, and say we will fund such an activity. I mean, you need it. Otherwise, it just loads another thing onto very busy programs. So here was something which we already do. So if, for example, I'm going to make this up, but we decided that fibrosis was an interesting state, somebody who had worked on that and studied could say, well, here's the essence of fibrosis, and this is it in the kidney, and this is it in the liver, and actually there's a whole nother one associated with surgical wounds, and they have the following similar different features, and you could do that in we can think what it is, 30-minute lecture, somewhere, that's still fairly short, and it would feel exactly the way we now teach flipped classrooms, right? And then if you're live to it, there could be a 30-minute discussion session, and if not, that gets archived. So I think it would look like what we had just done for the introduction, but it would be a little more prepared around a topic, and that would be a way to share learning. But I also think we would then almost just meet people in the process, both of and they could have more than one participant. In fact, the ideal one would have two people working together or two back-to-back. Here's what a pathologist thinks about fibrosis. Here's what a molecular person who works on mechanism of, receptor biology, for example, thinks. So that's sort of what I was thinking, is it both served, it had three features. One is it allowed us to learn about topics that we last sort of learned about in graduate school and have changed in the intervening 75 years. And the other one is that it would, to the very act of doing it, would potentially bring disciplines and groups together. But it would also have a concrete feel to it. It's something you could propose and see after a year if you'd done it or not.
Mm-hmm. I like that. I mean, I personally think obviously it takes investment from the folks preparing it, but these are people who are experts in that topic. And I think it would help, help bridge the gap for some of these questions. I would read about fibrosis and learn something new from hearing that, and how we might even think about modeling it. So I like that, and think we could push that out as well.
Well, the way we do it, coming back to, I think a little bit what Anna Maria was talking about, is that we do have resources, annotated resources of genes, right? I mean, interestingly, as those resources get bigger and bigger, they get harder and harder to manage, right? Gene cards gets less and less easy to understand, but we don't really have similar resources about cell types, cell states, excuse me, that span this. And, some of this would be reading the review literature. It's not as though nothing's known. In fact, a lot is known, and that's what the challenge. So this would be sort of a kind of expert trying to bring her opinion to bear and putting a resource together for the group.
Mm-hmm. And I'd be curious to see how this can really interact with the initiative that is underway to annotate cell types, right? That's a separate sort of definition. I think there was a whole working group dedicated to that, maybe led by Peter Karchenko and others. And I know that's happening with NIH-HCA and so forth, but it would be good to learn and not develop these cell state annotations in a separate framework from what's already being developed there. And I think, again,
and type and state, as Anna pointed out, are closely, sorry, Anna, they're closely related, so they would have to come together. Even in that case, it would be very interesting to see a sort of debate between people who are very prior knowledge driven. Let's say in immunology, you say, you know, for 50 years, we've studied T cells, here's what they are, or more than that, 100 years. And other people say, no, no, it should be purely data driven, and this is what we see in a UMAP, right? There's quite a debate going on in immunology there.
Yeah, no, I agree. I agree that it needs to be integrated. We don't want something completely, you know, not bound with what we have with the cell type, because it's been a huge amount of work in trying to define the cell type. And so I think that we want it together, but we need to discuss the approach, because I think there is where, you know, it's been one of the people who work in the cell ontology, and I know the states are kind of complicated, even for definition. That's why they are always not being put in there. But I've been involved in all the immune cell type in the cell ontology, but I know that there is a need of trying to define this state. So I think this should be a kind of collaboration between the two and trying to define a way that could be linked, but even not, you know, in some way linked, but able to represent what is needed, because what is needed when you do model, when you try to represent biology, is not always, you cannot only rely on the pure way of defining thing, because sometimes you need something that, you know, is more attached to some part of the reality that maybe is complicated to define correctly, but still is needed.
Hey, this is Jonathan. I have a comment in this regard, and I think I'm just underscoring what Anna it's just said, but also I heard someone else say earlier, you know, the usefulness of these is case specific. I think Peter touched on that, Ian touched on that, and Anna was just talking about that. But there may also be more than one. And so what seems to occur in my experience in using these ontologies is the ones that get used are the ones that are useful. I mean, it's sort of a circular argument, but I think having a use case in mind that we think affects a lot of people and making something that works in that case and then getting it used is really the goal. And it's not to cover the landscape and make an encyclopedia. I just don't, you know, I think we keep that goal in mind. There may be a couple that are quite close to one another and end up competing, and that's all right. And one of them will get used.
Yeah, that's a valid point. And it probably will take some resolution and more data and, you know, across context to really hone in and resolve that, or just to have, yeah, two related states, as you said. Excellent. Thank you everyone for this really robust discussion. I do hope that we can continue this thread, and your names are on here. Those of you that have engaged, if you haven't, it's posted in this box and you can please annotate it. Please do. This is a working group, and excited to see where this can go. I guess one question that I would just put to us before we disperse is, are people interested in setting up a time to discuss what we talked about here further around cell states? Do we want to fix a time? We're all on shutdown for some foreseeable future. Do we want to set a date in a month or something while it's still fresh to talk more about how we might follow up on this? Would people be game?
Sure. Yeah.
Yes, I think actually we're going to see, you know, I noticed the prep that I'm doing for coordinating a subsequent meeting, I think is going to come up with some of the same issues. So I think we'll probably, as we mesh the various things together, this will emerge, my speculation is, as an area of substantial interest.
Great, yeah. So Peter, hopefully you can also be a point person on that and feedback what comes from your next session and whether that overlaps, and maybe we can try and fix the date roughly, I don't know, a couple weeks from now, just to follow up and hash this out further. keep everyone engaged. That'd be great.
Yes, after the session today, I'll be in email touch with you.
Excellent.
And anyone
Christina,
else.
I put a post-it note on the soapbox sign up indicating we were interested.
Excellent.
Sean, you god, well done. Thank you.
Thanks, Cody.